All right, welcome to the second video from chapter 11, which is about enzyme catalysis. In this video, we're going to discuss serine proteases. So just as a little review, in the first video, we talked about how we define and categorize enzymes, and now we are going to the mechanism of a specific enzyme called serine protease, or a class of enzymes called serine proteases. We're going to go into the reaction that they help to catalyze, the structure of the enzyme active site, as well as the mechanism of this protease. So, first of all, let's talk about what are serine proteases. What do they do? As the name might suggest, protease, meaning cutting a protein, serine, right? So these serine proteases, their job is to cleave proteins, and they use within their active site a serine residue of a serine amino acid. And there are a number of different enzymes that have this type of serine protease activity. Some of these enzymes are digestive enzymes that help to hydrolyze our peptide bonds of proteins. Some of them are involved in coagulation of blood, where you also have the cleavage of proteins to help activate them. There are a lot of serine proteases also involved in inflammation and the inflammatory response. And there is also a relation of serine proteases to heart disease. And we won't get too much into these, but there is more information about them if you're interested here. So let's now talk about the digestive serine proteases, meaning the ones that are active along your digestive tract to help break down proteins in your diet. And so some examples of these are chymotrypsin, trypsin, and elastase. All of these digestive serine proteases have high sequence homology and structural similarity, meaning that they had a common ancestor that they all derive from, but that they have since diverged and now have some differences to them that we will talk about in a little bit. So there's some things that are very similar due to their high sequence homology and structural similarity. And in particular, they have the same residues that are working in their active site. And these residues in the active site are also called the catalytic triad because there are 3 of them that are helping to carry out this catalysis. There's an aspartate, a histidine, and a serine. And of course, the name for it comes from serine proteases or from this serine in the active site. So that's what makes them the same, is this catalytic triad. What makes them different are their different substrate specificities. And these different substrate specificities are due a lot to this specificity pocket, which is shown over here in this diagram on the right, which is sort of right next to this catalytic triad, which we have over here. And this serine protease is being shown here with a molecule right in that active site interacting with that catalytic triad and also being selected for by that selectivity pocket. So let's talk more about how that specificity pocket helps to choose a specific substrate, right? So the specificity pocket is specific for the amino acid that is right before the cleaved bond in that peptide or in that protein. And the differences in those specificities for amino acids are due to structural differences in that specificity pocket. And so we have on the right pictures of those 3 digestive serine proteases that we just mentioned, and pictures of each of their specificity pockets and what makes each one unique. So let's first start with chymotrypsin, which is shown towards the top. It has a fairly deep specificity pocket. And it is not charged. It has some glycines in it. And so this ultimately, especially because of its deep, large shape, chymotrypsin is good at selecting for bulky hydrophobic amino acids, such as phenylalanine, which is shown in this picture. And again, so phenylalanine will be the amino acid right before the peptide bond that's about to get cleaved. Different from chymotrypsin, we have trypsin all the way on the right. This one has a little bit of a shallower specificity pocket, and it's negatively charged with this aspartate on the bottom. And so this makes positively charged amino acid residues like lysine very favorable to sit in this specificity pocket. And so in this example for trypsin, the amino acid right before this cleaved bond, which is shown again here, this cis-sil bond, it's going to select for positively charged amino acids to sit in that specificity pocket. And then our last example is elastase. Excuse me, the positively charged elastase also has a pretty small specificity pocket. And there are sort of relatively bulky amino acids extending into this pocket as part of the enzyme. And so that makes the specificity pocket, in alanine. Wow, I cannot talk today. The specificity pocket in elastase is selective for small neutral amino acids, such as alanine, which is shown here. All right. So now that we've talked about the specificity pocket and how these types of serine protease enzymes choose their substrates, let's now really focus in on that active site and look at the steps in catalysis. So first we'll consider the enzyme substrate complex. And this is basically the beginning state. We have the enzyme, we have the substrate that's entered into the active site, but nothing has really happened yet. So let's look at the three different amino acid residues in our catalytic triad. So in the middle, we have our histidine, right? It's got this imidazole ring. And we've talked about in previous lecture how histidines are really great for acid-base catalysis because they fairly readily will donate and accept protons. And we'll see examples of that throughout this reaction that we're going to follow. So the nitrogen on the histidine has this lone pair, and it's forming a hydrogen bond with the hydrogen that's in this hydroxyl group in our serine as part of the catalytic triad as well. So the serine and the histidine are forming this really nice stabilizing hydrogen bond with each other. The other end of the imidazole ring in the histidine has a nitrogen with a hydrogen attached, and we have here another hydrogen bond. And this hydrogen bond is being formed with the aspartate. That's the third residue in our catalytic triad. So just a note here, aspartate's main role in this whole process is to help really stabilize the histidine's imidazole ring and to help orient it in space so it's able to properly interact with the substrate. Aspartate isn't going to directly interact with the substrate, but it is really important to help out this reaction to make it happen because of how it's helping to coordinate the other parts of the catalytic triad. As we'll see going forward, the other two amino acid residues here, the histidine and the serine, are both going to interact with our substrate. And our substrate, like the diagram shows, is a polypeptide. Here it's shown in this blue color. And specifically, we're going to focus on the peptide bond. That's this bond that joins one enzyme's amino group to one amino acid's amino group to another amino acid's carbonyl carbon, right? So this bond is the peptide bond, and this is the bond that's going to be broken by our serine protease. So first, let's look at how that happens. So when we started, right, we had this hydrogen bond in between the nitrogen in this imidazole ring and the hydroxyl group on the serine. The lone pair on that nitrogen is going to essentially take this proton away from the serine, leaving this serine in this essentially oxyanion form, and so that's going to the electrons here on this that were originally forming this bond, right, are now going to attack the carbonyl carbon that's part of the substrate, that peptide, that one end of the peptide bond, right, and then ultimately that's going to end up breaking this double bond, pushing the electrons up to the former carbonyl oxygen, and that's going to end up in this tetrahedral intermediate, where you have this formerly carbonyl carbon right here that is now being forced to make 4 bonds. We have this oxygen here that's forced into being negatively charged, and so it's this tetrahedral intermediate in the case of the substrate that is really relatively unstable. It's not very favorable, but our catalytic triad is helping to stabilize it, and it's doing that in a couple ways. So first we have our serine, right? It formed this nice covalent bond with the carbon, and that's going to help to sort of better maybe distribute the electrons than they would be without it there. We also have in our imidazole ring from that histidine, the proton that was originally part of the serine is now attached to that imidazole ring on the histidine and is able to form a nice hydrogen bond with the amino that's part of the peptide, the other end of this peptide bond, right, that we're going to break. So both sort of ends of the peptide bond are being stabilized by the amino acid residues that are in the catalytic triad. Meanwhile, aspartate is off to the side. It's doing its job. It's helping to stabilize this other end of the imidazole ring on the histidine, but it's not directly interacting with our substrate. So just as a brief interlude here, we have this oxyanion as part of our tetrahedral intermediate, and it is really unfavorable. Those electrons really want to go somewhere, and so we just talked about all of these different stabilizing interactions that are being formed with the catalytic triad, but the oxyanion has its very own special stabilizing interactions in addition to this, and this is called the oxyanion hole. So this is a slightly different view from where we were before, but so let's first orient ourselves. So we have our catalytic triad. Here's our aspartate. It's helping to stabilize this histidine with this hydrogen bonding, right? And then we have our serine. And so this picture on the left is showing the before part of the reaction. So we have this nucleophilic attack by the hydroxyl oxygen on the serine onto the carbonyl carbon that's part of our substrate. Right, here's our bond that's going to be broken right here. Apologize for drawing in red on red. And so the electrons get pushed up to this carbonyl oxygen, forming our tetrahedral intermediate that has this oxyanion. But conveniently, this negatively charged oxygen is right by these stabilizing parts of the amino acid backbone of the enzyme itself. So these amines that are part of the backbone of the enzyme are able to form these kind of hydrogen bonds with this oxyanion. That's the tetrahedral, that piece of the tetrahedral intermediate. So that's another way that this intermediate form is being stabilized by the enzyme, along with all those other stabilizing interactions that we discussed. So let's get back to our overall picture of what's happening during catalysis. So we have our tetrahedral intermediate, and this oxyanion doesn't want to stay for long. So the electrons are going to start to collapse back down, sort of kind of towards where they came from. So this electron pair goes back to reform this double bond, to reform the carbonyl. But instead of pushing off the serine, instead we have the breakage of this peptide bond when these electrons go over towards the amino nitrogen, helping to take this proton off of the imidazole ring, and the imidazole ring is going to take back its lone pair. So now we've changed the bonding slightly of this hydrogen that we just talked about, and we broke this peptide bond. So ultimately now we have this new intermediate, this acyl enzyme intermediate. And so now we have this cleaved substrate, right? This peptide bond is gone. I don't even want to draw anything there. And so now we have this cleaved amino end, right? It's only hydrogen bonded to this histidine that's part of the catalytic triad. It's able to just leave the active site when it's ready. It's not covalently bonded to anything in the enzyme anymore. But we do still have covalently bound to the enzyme is our new C-terminal end, right? We have this covalent bond that the carbonyl carbon has formed with our serine that's part of the catalytic triad. So for the enzyme to reset itself and also to detach the other half of this protein, more things have to happen, right? So what happens next? Next we have, like I mentioned, this release of the amino terminal product, and we instead, now that floated away, now a water molecule is able to kind of float into the area where this catalytic triad is. So now, before we had hydrogen bonded, we had our N terminus of our new polypeptide chain, right? It was hydrogen bonded to this nitrogen in the imidazole ring of our histidine in our catalytic triad. When water flows in, it's going to replace it. So now the hydrogen atom on the water is now hydrogen bonding with this nitrogen atom on the imidazole ring of this histidine in the catalytic triad. And ultimately, this is going to help us reset our enzyme. So let's see how that works. So we have, again, another nucleophilic attack from the lone pair of electrons on this nitrogen in the imidazole ring. It's going to form a covalent bond with this proton, and now this hydroxyl group, the lone pair on this oxygen, is now, or from this bond really, right, is going to attack the carbonyl carbon, again forcing these electrons up to the oxygen, forming yet another tetrahedral intermediate that has yet another oxyanion. And so this looks a lot like our other tetrahedral intermediate, except this sort of side is no longer the other half of our protein, right? It's just the end of a water molecule, just an OH. So now we have the same reversal that we had the first time we had our tetrahedral intermediate, where these electrons collapse back down to reform that carbonyl. And this time now we are going to break our bond to serine. So these electrons are now able to go over to this proton that's part of the imidazole ring, ultimately helping to reform the hydroxyl group on the serine that is hydrogen bonded to this nitrogen that's on the imidazole ring of that histidine. And because we broke this bond to the serine, we're able to now release our new C-terminal end of that original protein that we were cleaving. And now the catalytic triad is back to where it started, right? Aspartate is still stabilizing our histidine imidazole ring with this nice hydrogen bond. The other nitrogen on the histidine imidazole ring is hydrogen bonding with the hydroxyl group on the serine. So everything is back to where it started, and this serine protease is ready to cleave a new substrate. So let's summarize what just happened. So for serine proteases, they all have a specificity pocket that helps to them be very specific to which amino acid can be right before the cut, right before that peptide bond that's going to get cleaved. And our three examples were our digestive enzymes, our chymotrypsin, which has a specificity pocket that selects for bulky hydrophobic amino acid residues, trypsin, which selects for positively charged amino acid residues, and elastase, which selects for small neutral amino acid residues. And again, these are before the peptide bond that's about to get cut. The part of the enzyme in the active site that's actually responsible for catalysis is the catalytic triad, and that consists of those three amino acid residues, histidine, serine, and aspartate. And we talked in a lot of detail about how histidine is responsible for a whole bunch of acid-base catalysis, the serine is responsible for covalent catalysis, and the aspartate forms very important hydrogen bonding and orientation cues for the imidazole ring in that histidine. And we also took some time to discuss the oxyanion hole, which helps to stabilize the tetrahedral intermediate, which, as we hopefully know, forms an oxyanion. So with that, now you are ready to answer the Chapter 11 serine protease practice questions.